All right, so the picture on the left is more gene expression. So this second part, we talked about the operons. What I want you to realize is it's a mess. It is difficult, lots of little details. And so what I want to do is real quick overview. Don't want you to get lost in the details, but I just want you to realize how many little adjustments can be made on top of making proteins to make this work. So what we want to do is look at a few of those. One is the chromatin structure. And so what you're going to hear about later, and I'm not going to do it much now, but there are some words that you should just associate with. Again, in BioA, they should talk a lot about this, but you have acetylation, adding an acetyl group to an amino acid in a histone tail. So they have these little histone tails sticking out, and when you add these little, when you add the acetyl groups to them, they physically change the chromatin structure. And by doing this, it promotes transcription. It makes it easier for you to transcribe. So by adding these acetyl groups, it actually makes this function better. On the other hand, you can actually inactivate it by adding methyl groups to the cytosine. Cytosine seems to be the nucleotide that it attaches to, and this will actually shut it down. So what I'm trying to give you here is these two little processes at the chromatin level will change the structure and make it either easier or stop transcription. So think of adding the acetyl. A is promoting transcription, and then the M is actually slowing it down. Another thing that we realize is it's not all about exactly what the code is. It turns out it's also what's inherited. It's basically environmental conditions that organisms are exposed to. So on the left are two mice. They're being fed the same diet. They have the same genes basically, but the one on the left had different conditions that it was exposed to inside mom, and it does not break up fat as well. So you end up with a fatty mouse. So you know, you can go, if you're overweight, you can go home, blame your mother. Don't do that. And then on the right hand side, we have some people who were starved to death. And so what happened, diet of mom again had an effect on the children, again, not because of what the chromosome said, but because of the unique situation mom was put in. So epigenetic inheritance. All right, regulation of transcription initiation. Again, what we want to make sure you understand is there are these general transcription factors that tell it whether to run. Without the transcription factors, RNA polymerase won't attach. And then there are some things called enhancers that increase the chance that you're going to get these things to work, and you're going to get transcription happening more. And there's actually even specific transcription factors with enhancers. So what we mean by specific versus general means it'll just run. The specific transition factors might determine what actually happens in that cell. Because what happens, again, what you got to remember, and this is the part that's a little hard for people, I think, to understand, is each cell has all this information. So it's got to figure out what actually am I turning on and off in each cell, because I don't want my liver cells building lenses that would be in the eye and vice versa. So you really need to have these very specific transition factors to turn on particular parts of the DNA strands. And then this is a little more vague, but we're looking at nuclear architecture. And so here we're looking at these loops and how they attach to each other. And we know that chromosomes come together and we we know during like mitosis and meiosis, these things condense and they take a particular shape. And what we're finding out now is that shape is extremely important. We've always known that shape is function. But these chromatin loops and transcription loops and territories all kind of interact together on that final product. So we're finding now the nuclear architecture, the things that are happening inside there, we just assumed were always the same. And now we know there are tiny differences, and those differences can have a huge effect on how things are expressed. And then some of the post mechanisms. So after it's been transcribed, what can happen? Again, RNA processing, we talked about the 4 steps. But we didn't talk about is it is possible to have different exons, and depending the spliceosome will splice together different ones in different places. Okay, so here we have 1, 2, 3, and 5 being expressed. Here we have 1, 2, 4, and 5 being expressed. Same basically messenger RNA. The spliceosome decides how that's going to be put together. And so we're working on how is that controlled. The other one is initiation of translation. How does it start? Again, that's transcription factors. We're finding out more and more. We're still very vague on this, but each factor, we're trying to figure out how these work, how it's decided what factors involved. So this is all kind of at the biochemistry level. Messenger RNA degradation. It turns out there are signals now that are being said on how long the messenger RNA actually lasts. Remember, I told you the polyribosomes and how many you can go through. That will control how much of the protein is made. A lot of protein, a little bit of protein, and again, depending on what that protein is it an enzyme? Is it a building block? Some kind of construction, physical structure? All of those things are important. And then the last one is protein processing and degradation. It turns out even the proteins themselves on how long they last. There's actually an enzyme called ubiquitin. It's a protein. And when that's around, that this will actually destroy proteins. Apoptosis, or how long a protein can actually last, maybe an enzyme. So what we're trying to do with this is just show you how complicated this system is. How many different ways that these cells can be manipulated into doing things. Okay, and then the last one is non-coding RNA. There's pieces of RNA that aren't being read, and there's microRNAs. miRNA is what it's called. These little small single-stranded RNAs that can bind on the complementary sequence, and by doing that, it can block things. And when it does that, it means the mRNA can be degraded or blocked. You have small interfering RNA, similar to above, but it's a slightly different precursor. So micro and small kind of do the same thing. It's just a matter of where they come from. And then even non-coding RNA can actually bend chromatin and make it slightly different and make it less functional or more functional, depending on how it's bent. So again, I'm just trying to hammer home structure, function, and all these little things that are involved that we did not realize that can actually determine what is actually physically expressed.